Felous, and today I'm going to talk about the problem particulate chromate exposure does not inhibit homologous recombination repair in North Atlantic right whale, Eubelina glacialis. And prolong means taking for a long time, and exposure means stay in a kind of environment. And inhibit means to stop or reduce a kind of reaction. And first we introduce Eubelina glacialis, which is North Atlantic right whale, and is now in danger because the ship strikes these features and harmed by fishing gears, these two pictures. And maybe there was more potential risk factor that environmental contaminants, and which means environmental pollutants. And these pollutants is particulate hexavalent chromium, and it's well known in our daily life for preventing metal from getting rusty. And hexavalent means 6, and which means the chromium can bind to 6 chemicals. And why is this pollutants will influence the right whale? This is the left picture. This brown region is where the water resource is already polluted by the chromium, the hexavalent chromium. And the right picture is the river in America. You can see the river will carry the pollutants and bring it to the North Atlantic Ocean, where the right whale active. So we might think that is a kind of potential risk may affect right whale. So how does these pollutants affect on the right whale cells? It will cause the cell's chromosomes instability, which means chromosome unstable. And in a cell, if you zoom in, this is chromosome where the DNA is stored. So when the chromosome becomes unstable, it will cause the cell to get cancer. And what is homologous chromosomes? Homologous chromosome, every homologous chromosome is a pair. One of them is from your father's chromosomes, and the other one is from your mother's chromosomes. And when your cell is growing, it has to divide from 1 into 2. During this process, we call this process meiosis. And in this stage, all homologous pairs have to line up like this, and we call this stage metaphase. And during this stage, during metaphase, because all the homologous pairs are lined up together like this, and when the chromosomes is already instability, already unstable, it will cause the part of DNA sequence will exchange, and we call this situation sister chromatic exchange. And it might also happen the part of DNA sequence will broken like this, will break like this, and we call this plastogen. But I will just name it DNA damage later. And the cells have to repair the broken DNA, and we call this repair homologous recombination repair, which is HR repair. And it's a kind of protective DNA repair, and you can use the healthy DNA to repair the broken DNA with RAD51. Then what is RAD51? It's a part of DNA sequence that can code this kind of protein, and they are related to HR repair process. And if we get to know the RAD51 response, we can know the cells is working on the HR repair or not. So we already know the high pollutants in the public water resource, and it causes human to get lung cancer because this kind of pollutants will inhibit the HR repair and make the cells to get cancer. So in previous research, they already know right whale has less DNA damage than humans. And one of the research found found that more prolonged exposure time inhibits HR repair on human cells. This is human situation. But how about right whale situation? Will more exposure time affect on right whales on cells? We already know from the previous research less DNA damage right whale will have less DNA damage. So in this research, we want to prove that more prolonged exposure time does not inhibit HR repair. So we collected cells from stranded right whale, and we bring the sample back to the laboratory and control the temperature in 33 degrees Celsius. And we separate them into two groups. One is 24 hours, and the other one is 120 hours. And then we add zinc chromate as hexavalent chromium, and then we store the sample in metaphase situation, and then we add DAPI. This chemical can help us mark RD by 1 and make it glow in the dark. Will help us more easy to observe. And this is our first result. The x-axis is zinc chromate concentration, and the y-axis is relative survival. You can see with concentration increasing, the more cells dies. And the black line is 24 hours, and the gray line is 120 hours. You can see the difference between them is not very significant. So we say more prolonged time does not significantly decrease the relative survival. And the second result is x-axis is zinc chromate concentration, and the y-axis is percent of metaphase with damage, and this is total damage. And black one is 24 hours, red one is 120 hours. See the concentration increasing, and the cell damage will increase too. So we say more prolonged time does not increase damage. Oh, the difference between the exposure time. And the third result is this is zinc chromate concentration, and this is percent of cells with more than 5 RD by 1 FOSSI. And then what is FOSSI? FOSSI means a point, and there are many RD by 1 together in this point, and we call this point FOSSI. And if there is more than 5 FOSSI, and it will appear on this data. You can see 24 hours black line is more than 120 hours. And in this picture, this is RD by 1 response, and this is after adding DAPI chemical. Yes, and merge means combine these two pictures into this picture. And darker pictures means more RD by 1 FOSSI. So in this picture, both of them represent the 24 hours is more than 120 hours. So more prolonged time decreased RD by 1 FOSSI. And the fourth part, this is sister chromatin exchange. You can see in this picture, there was part of DNA sequence exchange. And x-axis is exposure time, and y-axis is SC per metaphase and SE per chromosomes. The different colors means different zinc chromate concentration. And between these two exposure time, 24 hours and 120 hours, their results are not significant. So we say more prolonged time does not increase sister chromatic exchange. So we made a conclusion. With relative survival and sister chromatic exchange have no significant result. But in DNA damage and RD by 1 FOSSI, they have higher value in 24 hours. And in this different exposure time, they both have RD by 1 response. But only in 120 hours have less DNA damage, and which means there was more broken DNA in 120 hours have been repaired, and because in 120 hours it gives cells more time to repair the broken DNA. So the result, we can know the prolonged time does not inhibit HR repair in right well-done cells, and it even have higher efficiency in 120 hours. So right well have higher stability of chromosomes and then have higher efficiency of HR repair ability. So maybe in the future, if we can enhance the HR repair response in human cells, and we can know how the HR repair response work in the right well cells, and we can help our human anti-cancer improve our technology. And this is the end of my talk. Thanks for your attention.